Hello everyone, welcome back to the session. So we are discussing about the CUET PG exam in the zoology. Okay, so in zoology we have started the cell biology chapter. So this is the fourth day of the cell biology. So in today's class we are going to discuss about the nucleus as well as the chromosome. Okay, so far we have completed the cell theory and what is the difference between prokaryotic and eukaryotic cell, and in yesterday's class we have discussed about the plasma membrane, and in today's class we are going to discuss about the nucleus and the chromosomes. Okay, so what is nucleus? Okay, see, this is the cell. Okay, so inside the cell we are having a nucleus. Okay, so in yesterday's class we have discussed completely about the plasma plasma membrane. Okay, so now in the plasma membrane, so which is the center part of the cell is called as a nucleus. So you should know whether nucleus is a double membrane or the single membrane. It is a double membrane, the first thing. Okay, so it is a double membrane bound layer, double membrane bound organelle that houses a genetic material and it regulates the cellular activities. Okay, so this is very important. The first important thing will be the nucleus will be the double membrane bound organelle. Okay, so double membrane bound organelle. So in the nucleus we are having a genetic material and we know that in human beings, and the nucleus contains a genetic material, and that genetic material will be the DNA, deoxyribonucleic acid. Okay, and this is the one regulating the cellular activities. Okay, so the nucleus was first discovered by a scientist called as the Robert Brown. Okay, Robert Brown in 1831, and it is present in the eukaryotic cells and it is absent in the prokaryotes. They possess a nucleus. So in yesterday's class I have explained you what is the difference between nucleus and the nucleus. You should not think that nucleus and the nucleus are similar to each other. Okay, it is different. Okay, so in prokaryotes they are not having a proper nucleus within it. Okay, so the area, the region where the genetic material present in prokaryote, it is considered as a nucleus. But here in eukaryotic cell, in a cell, that cell contains a nucleus with a proper double membrane bound organelle. It is a double membrane bound organelle. Inside that membrane we are having a genetic material, and that is a DNA. It was discovered by a Robert Brown. Okay, now, so how can we expect in the question? Okay, so this nucleus act as a genetic, structural, functional control center of the cell. Okay, this nucleus is a genetic, structural, functional control center of the cell. Okay, see, just say for an example, for a computer, CPU is the brain of the computer. So like in the same way, nucleus is considered as a structural and functional control center of the cell. Okay, so this is what a structure of a nucleus. See, in the nucleus we are having, see, we are having an outer membrane and the inner membrane. This outer membrane and inner membrane together we are calling them as a nuclear envelope. Okay, so this nuclear envelope, you should not think this as an outer membrane or inner membrane, both outer membrane and inner membrane. So this nuclear double membrane is called as the nuclear envelope. Okay, now this nuclear envelope, they are having a protein. That protein is called as a nuclear lamina. Alright, the nuclear lamina, so see here, this is a cell and this is the nucleus. So here we can see the cytoplasm. Okay, now the center part, this is the central part. It is like this. The central part, they are also having a gel-like structure. That is also, they are having a space. So that space is called as a nucleoplasm. Okay, now inside the nucleus we are having a chromatin. Okay, you should know what is mean by chromatin, what are all the types of chromatin, what is mean by chromosome, what is mean by chromatin reticulum. Okay, so what is mean by chromatid, all these are different. Okay, you should not think all these were similar, but here let me explain you what is chromatin in the upcoming slides. Okay, so here we can see the chromatin, and inside the nucleus we are having an important area that is called as a nucleolus. Okay, so the nucleoli are involving in an important synthesis. We are going to discuss about that. And the nuclear envelope, we are having a pores, lot and lots of pores within it. Approximately 3000 pores can be seen in the nucleus. So that is why we are calling them as a nuclear pore. And also we are having a ribosomes. So these are all the structure which can be present in the nucleus. Okay, so now, so the nucleus, what is the difference between the nucleus which is present in the prokaryotes and in the eukaryotes? So in eukaryotes, true nucleus will be present. In prokaryotes, no true nucleus will be present. As it is not a true nucleus, we are calling this particular segment as a nucleoid. Okay, so anucleate cells. Anucleate cells. So this anucleate cells, they are having a mammalian RBC. So what is meant by anucleate cell and nucleate cell, multinucleate cell? See here. Nucleate cell means a cell contains a nucleus. That is what a nucleate cells. Anucleate cells means A means absence. A nucleus is absent in the cell. So that is what an anucleate cells. Multinucleate cell means one cell has many nucleus. That is what a multinucleate cells. Okay. Do we have all these three types of nucleus? Yes, of course we are having all these three types of nucleus. Okay. Majorly we are having uninucleate cells. All cells having a nucleus. This is an important thing. And another thing will be, so the cells which is not having a nucleus, that is called as an anucleate. Of course, we are also having an anucleate cells, that is called as an RBC. RBC doesn't have nucleus in human. Okay, so why it is not having a nucleus? Because RBCs needs more space. Okay, RBC has a hemoglobin, and that hemoglobin requires more space to carry the oxygen. So that is why, to increase the space, there is no point of having the nucleus within that. Okay. Now, multinucleate cells, so cells which is having many nucleus is called as a multinucleate cells. Do we have in human beings, do we have a multinucleate cells? Of course, we are having a multinucleate cells. In the first class, we have discussed about the syncytium and sinusotic hyphae. Okay, syncytium means the cells which is having many nucleus. Okay, so fusion of nucleus from many cells. That is what we can we can see in the skeletal muscle fibers. Okay, and in some fungi there is no cytokinesis, but only karyokinesis will be taking place, but not a cytokinesis. So if there will be no process of cytokinesis, we are calling them as a sinusotic hyphae. Okay, that will be seen in some fungi. Okay, now see, this CUET exam often asks these exceptions, that is the multinucleate cells and the anucleate cells. Okay, all other cells having the uninucleate. Okay, uninucleus. Now, what are all the structure of the nucleus? In the structure, what are all the most important structures? Okay, see here, the nuclear envelope. Nuclear envelope is nothing but a nuclear membrane. Okay, so in the nuclear membrane, as we have studied before, it is a double membrane bound layer, membrane bound organelle. So in the double membrane structure, we are having an outer membrane and an inner membrane. So in the outer membrane, so this outer membrane of the nucleus is continuous with rough endoplasmic reticulum, may bear the ribosome. See here, so this is a nucleus. So this nucleus will be surrounded by a nuclear membrane. So we have studied this nuclear membrane has outer membrane and the inner membrane. So in the outer membrane, so this is the one continuous with the rough endoplasmic reticulum. Okay, so what is meant by rough endoplasmic reticulum? So in the endoplasmic reticulum, if ribosomes is present, we are calling them as a rough endoplasmic reticulum, which is involving in the protein synthesis. Okay, so if the ribosomes are absent, we are calling them as the smooth endoplasmic reticulum. Right? Now, see here, in the inner endoplasmic reticulum lines the nucleoplasm. The space inside the nucleus is called as a nucleoplasm. Now see here, this is an outer membrane and this is an inner membrane. Between these two membranes, we have seen the space. That space is called as a perinuclear space. Okay, and the space, what is the distance? 10 to 50 nanometer between the outer membrane and the inner membrane. Okay, now what will be the function of this nuclear envelope? So as we have studied before, so this nuclear envelope having these pores. Okay, through these pores, all the contents which is present in the nucleus will be entered into the cytoplasm, and from the cytoplasm it will be entered into the nucleus. All these things have been mediated by the nuclear pores. This is one important thing. See here, the function of the nuclear, and why the nuclear why the genetic material has to be covered with a nucleus, and what is its why it is necessary. See, all the nuclear contents has to be separated from the cytoplasmic content, so it is acting as a separation. It is regulating the exchange of materials from the cytoplasm to nucleoplasm, from the nucleoplasm to the cytoplasm. Okay, so this is the function of the nuclear envelope. Now, so we have studied the nuclear pores are present in the nuclear envelope, but yes, nuclear pore is present in the nuclear envelope, and you should know what is exactly present in the nuclear pore and what is meant by nuclear pore complex. Okay. So nuclear pore complex, again it is a protein, a large protein complex embedded in the nuclear envelope. Okay, so this is a nuclear pore is nothing but a large protein complexes that is present in the nuclear envelope. So this pronuclear pore allow selective and bidirectional transport. Okay, so this is a selective and bidirectional transport. The nuclear pore will not allow all the components which is present in the nucleus to cytoplasm and cytoplasm to nucleus, but it is selective. Okay, and it is bidirectional. It is not a unidirectional, that is from the nucleus to the cytoplasm, but not from the cytoplasm to nucleus. It is not like that. Okay, it is mediating the transport from the nucleus to cytoplasm and cytoplasm to the nucleus. It is a bidirectional transport. Okay. Now, what are all the things are transmitted through the nuclear pore complex? That is the smaller molecules and the larger molecules. Smaller molecules doesn't need any of the help. Okay, it is simply diffused through the envelope. So yesterday's class we have discussed about the plasma membrane. So in that whenever we are discussing about this plasma membrane, we have studied about the active diffusion and passive diffusion. Okay, so in the passive diffusion, those organisms which are very small can be easily diffuses through the envelope, so that doesn't need any energy. So that is what a passive diffusion. So the larger molecules like proteins and RNA, they definitely need, they are definitely in need of an energy. So that's why we are saying that as an active diffusion. So if it is an active diffusion, definitely there has to be this nucleus are in need of the signal. Okay, so that is what we are calling them as a signal mediated transport. Okay, signal mediated transport. Okay, so nucleus need a signal to send something, to receive something. Okay. Now see here, so nuclear pore is not an empty hole, but instead it is a regulated gateway. As I have mentioned before, it needs a signal to receive and to send. Okay, so that is what a nuclear pore complex. Now what is mean by nucleoplasm? Nucleoplasm is a semi-fluid matrix which is present inside the nucleus. Okay, just like a gelatinous substance which is present in the cell, that is cytoplasm. The nucleus also having a semi-fluid matrix, that is what we are calling them as a nucleoplasm. What is exactly contains in the nucleoplasm is, the nucleoplasm is the one storing or containing this chromatin, nucleolus and the enzymes for DNA replication and the transcription. Why the enzymes for DNA replication and the transcription will be present in the nucleus? Why not in the cytoplasm? Because DNA replication and transcription will be taking place in the nucleus in case of eukaryotes, not in case of prokaryotes. This is an important point. Kindly make a note of it. Okay. Now, nuclear lamina. Okay, so what is mean by nuclear lamina? So it is made up of a lamin protein. Okay, so protein meshwork under the inner nuclear membrane. Under the inner nuclear membrane, we are having a protein meshwork. Okay, so that is what we are calling them as a nuclear lamina, as it is made up of a lamin protein. Okay, so what is the point of having the nuclear lamina under the inner nuclear membrane? Means it will be providing the mechanical support. Okay, it is involved in the chromatin organization. It is involving in the chromatin organization. Okay, now what is the structure of the nucleolus? What is the structure of the nucleolus? This nucleolus, the structure, it will be dense. Okay, so where this nucleolus is present? In the nucleoplasm. Okay, so now the structure of the nucleoplasm will be spherical and it is dense and it is non-membrane bound structure. This nucleolus doesn't have any of the bounded membrane. Okay, it will be free. and appears during the interphase. Okay, this nucleolus appears during the interphase, disappears during the mitosis. What is interphase, what is mitosis, everything will be discussed detailed when I explain the cell cycle and the cell division. Okay, so only if I explain in that place, it will be it is suitable. Okay, what is the point of having the nucleus in the nucleoplasm and what is its function? So first we have studied about where it is exactly present, that is in the nucleoplasm, and how its structure will be, and now what is the function of the nucleolus. Okay, so where exactly the rRNA synthesis, okay, ribosomal RNA synthesis and ribosome assembly will be happening in case of nucleolus, in place of nucleolus. Okay, see how in exam how can we expect a question? Nucleolus is associated with the ribosome biogenesis, not protein synthesis. You should think that it is not exactly where the protein is getting synthesis. Okay, the protein is not synthesis in the nucleolus, but instead you should know ribosome biogenesis. Ribosome synthesis will be taking place in the nucleolus, not protein synthesis. Okay, now what is the function of the nucleus? Okay, see here, function of the nucleus is not nucleolus, functions of the nucleus. First, it is the storehouse of genetic material, that is DNA. Okay, that is where we can find the DNA replication. DNA replication will be taking place in the nucleus. Okay, and transcription, that is RNA synthesis, will be taking place in the nucleus, and regulation of cell cycle regulation will be taking place in the nucleus. Okay, now control of cell growth and the differentiation, cell growth control and differentiation, everything will be regulated in the nucleus. Okay, so if there will be no nucleus, loss of cell division capability. Okay, see in RBC we have studied there is no nucleus present in the RBC, so that is why we are calling this RBC as an anucleated cell. Okay, so if there will be no nucleus, there is no point of having the cell division here. Okay, so this is very important. Now, what are all the things which are happening, which is transporting in case of nucleus? What are all the transport molecule and what will be the transport type? Okay, so if it is an ions or smaller metabolites, it doesn't need energy, it simply diffuse passively, so that is why it is coming under the passive diffusion transport. Okay, now if it is an mRNA or tRNA, that is messenger RNA and transfer RNA, it is a larger molecule, definitely it needs energy, so that is why we can classify them under the active transport type. Okay, now nuclear proteins, so if any protein, okay, nuclear protein has to be sent out from the nucleus or sent inside the nucleus, definitely it needs signal. For example, RAN GDP. Okay, so that is what very important, that needs a signal mediated import, signal mediated export. Okay, now this transport depends on the nuclear localization signals. Another thing, all these transport will be required are based on the nuclear localization signals only. Now, what is the difference between prokaryotic nucleoid and eukaryotic nucleus? See here, the nucleoid will be the membrane. Okay, so nucleus is a double membrane, but instead this nucleoid doesn't have any of the membrane, so the membrane is absent in nucleoid, and the DNA which is present in the nucleoid is circular and naked, but here in the eukaryotes it will be linear and they are histone bound. Okay, so now nucleolus will be absent in the nucleoid and it is present in the nucleus. Transcription and translation are coupled in the nucleoid in case of prokaryotes, and it will be separate process in the nucleus in the eukaryote. So that is why in the first class I have mentioned you prokaryotic nucleoid is not equal to eukaryotic nucleus. These are all the differences. Now, in exam, how can they confuse us? Okay, see here, nucleolus has a membrane. This is a false statement. Nucleolus doesn't have any membrane. Okay, nuclear pores allow free passage of all molecules. Okay, it is not all molecules, some molecules. Those are all in less molecular weight. So the statement is false. Heterochromatin or junk DNA are false. So this is a false statement. We are going to discuss about what is the heterochromatin. And prokaryotes have nucleus. This is false. Prokaryotes have nucleoid, not nucleus. So this is a false statement. Okay, so now this is a one liner. Nucleus discovered by the Robert Brown. We know that nucleus is discovered by the Robert Brown, and the nuclear envelope is a double membrane bound layer organelle, and nucleolus is responsible for rRNA synthesis and ribosome biogenesis. And euchromatin will be transcriptionally active. Let me explain you what is heterochromatin and euchromatin in the upcoming slides. Okay. And nuclear pores regulate nucleocytoplasmic transport. Why it is called as a nucleocytoplasmic transport? As it is bidirectional. Because all the contents, the nuclear pore will transport the contents from the nucleus to cytoplasm and from the cytoplasm to the nucleus. Both the way are possible. So that is why we are calling this particular transport as a nucleocytoplasmic transport or bidirectional transport. Okay. So that's all about the nucleus. Now we have to discuss about the chromosomes. Okay, so who discovers the chromosome? So the chromosomes are the thread-like structure that is present inside the nucleus in the eukaryotic cells. All these are an important statement. What is the structure? It is a thread-like structure where it is present inside the nucleus, and in case of prokaryotes or eukaryotes, it is in the eukaryotic cells. Right? Now, it was first reported by whom? That is Strasburger in 1875 in eukaryotic cells. Right? Now, the word chromosomes were introduced by Waldeyer in 1888. Now, who is Bridges? Bridges is the one in 1916 proved that chromosomes are the physical carrier of a gene. See here, we have studied about the nucleus. Okay, this is a cell. Inside the cell we are having a nucleus, and this is the nucleus. Inside the nucleus we are having a nucleoplasm where we can see the chromatin. Okay, so this is what a chromosome. Inside the chromosome we are having a genes. So Bridges is the scientist proved that chromosomes are the one carrier of a genes. This is an important thing. Okay. Now, chemically, chromosomes are composed of DNA and associated proteins. Okay. So, if you want to define what is chromosome, chromosome contains a genetic material, that is DNA, and the gene carriers, which means gene carriers and the genetic material together we are calling them as a chromosome. So, chemically, chromosomes are composed of DNA and associated proteins. Here, these associated proteins is nothing but an histone proteins. Okay. Now, what are all the chemical composition of chromosomes? The chromosomes are made of the chromatin. Okay. Made of chromatin. This chromatin consists of, what chromatin consists of? Okay. So, the chromatin consists of DNA, proteins, that may be either histone proteins or the non-histone proteins, and some small amount of RNA. See here, the chromosomes are made of many chromatin, and each chromatin has DNA and small amount of RNA and proteins, and that protein is histone proteins and non-histone proteins. Okay, so this is what a chromosome is. So now what is chromatid? So what do we have discussed here? This is chromatin. Okay, and you should know what is chromatid. Okay, see here, this is a chromosome. Okay, this is one chromosome. Okay, no doubt in that, this is one chromosome. So in that one chromosome, we are having two sister chromatids, which means like this. Okay, so this up and down, two identical sister chromatids. So one chromosome, we are having two chromatids. Okay, so why we are calling them as a sister chromatids? As it is present in the same chromosome, we are calling them as a sister chromatids. So you should know that one chromosome has two sister chromatids, that is in identical. So what does mean by non-sister chromatid? This is one chromosome and this is another chromosome, and this is the non-sister chromatids. Okay, so this is another important thing. Now, see here, the central point where the sister chromatids are joined with each other, that is called as a centromere. Okay, centromere is the one holding the true chromatids of the chromosome. Okay, that is a joining point. And during the cell division, this chromatids condenses to form well organized chromosome with definite size and definite shape. So this chromatids undergo condensation process during the cell division. Why this chromatids undergo a condensation process? To ensure a definite size and shape. Okay. See here, this is what a structure of a chromatid chromosome. So in the chromosome, this is what one chromatid and this is another chromatid. So two identical chromosomes. Okay, so two identical chromatids. And the short arm is called as a P arm. Okay, and the long arm is called as a Q arm, and the central point where the chromatids are holding together is called as a centromere. Okay, so this is where we can see the gene. That is what a DNA molecule. Okay, so the DNA molecule is a long string-like DNA molecule formed into a compact structure by proteins, which is called as a histone. So these are all the structure of the chromosomes. Okay. Now, a typical chromosome has constricted regions. So far we have studied about what is chromatin, what is chromatid, and what is chromosome, and what is centromere. What is P arm, what is Q arm, what is long arm, and what is short arm. Everything. What is mean by DNA, who is a carrier of gene. All these things have been cleared. Okay, now you should know what are all the constrictions present in the chromosomes. We are having two major constrictions. One is primary constriction and secondary constriction. Okay, so what is the primary constriction? See, we have studied the center point is called a centromere, where the two sister chromatids are holding each other. Okay, see here, that centromere is called as the primary constriction. Okay, so this is the one holding the two sister chromatids together. Now, this bears a specialized protein complex. See, so this centromere having a protein complex, all these protein complex together we are calling them as a centromere. And what is the name of the protein complex? That is called as a kinetochore. Okay, now, so this kinetochore is a proteinaceous in nature, and this is where the spindle fiber attachment during mitosis. During mitosis process, the spindle fibers is going to form. And why the spindle fiber is going to form? To separate the two sister chromatids of the chromosome. To separate, the spindle fibers is going to form, and for that the spindle fibers has to attach with the chromosome, where it is going to attach particularly in the kinetochore, which is proteinaceous in nature. We are calling them as a centromere. Okay. Now, see here, kinetochore is essential for chromosome movement, and it is right statement. We can expect this in an exam. Now, what is meant by secondary constriction? See, the secondary constriction present in some chromosome in addition to the primary constriction. Why it has to present? So, this is the region where the nucleolus is developing. Okay. So, we have studied in the nucleoplasm, we can see the chromatin, we can see the nucleolus. Okay, so the nucleolus will develop from the secondary constriction. Okay, so the regions, the nucleolus, okay, so where the nucleolus develop is called as a secondary constriction. Okay, but in that region, that region where the nucleolus develop is called as a nucleolar organizer regions. Okay, so this nucleolar organizer regions contains the ribosomal rRNA synthesis. That is where the nucleolus present. Okay, so this is an important point. Okay, so besides this primary and secondary chromosome constrictions, you should know what is meant by satellite chromosome, that is SAT chromosome. See here, a small chromosomal segment separated from the main body by a secondary constriction. Okay, through the secondary constriction, a small part of chromosome segment has been separated from the parent chromosome, and that satellite chromosome is called as a SAT chromosome. Okay, morphologically distinct in certain chromosomes. Okay, so we are going to discuss what will be the function of the SAT chromosomes. Now before that, what is telomere? So another very important structure of the chromosome will be the telomere. Okay, see, telomere will be the terminal end of the chromosome. End part of the chromosome is called as a telomere. And why we have to, why we need a telomere? So this telomere is the one responsible for providing the chromosome stability and prevents the chromosome fusion. See here, what is meant by chromosome? This is one chromosome and this is another chromosome. This chromosome will fuse with each other. This prevention of fusion is done by the fusion is preventing with the help of this telomere, which is present in the terminal end of the chromosomes. Okay. And this telomere contains a repetitive DNA sequences, and that is nothing but TTAGGG. Okay, so repeated many times. Okay, so in humans and the neurospora crassa, which is a fungi. Okay, so the telomere we have studied. This telomere has repetitive DNA sequences, and what is that repetitive DNA sequences is TTAGGG in case of humans and neurospora crassa. Okay, so these are all very important statements. Okay, now what is the function of the telomere? So here this telomere is the one preventing the fusion of the chromosome. So one chromosome will fuse with another chromosome. That fusion is preventing by the chromosome enzyme. Fusion is prevented by the telomere. Now this is the one protecting the chromosomes from degradation. So if one chromosome is fusing with another chromosome, the chromosome may lose its integrity and it may leads to degradation. Okay, so that chromosome stability, integrity will be ensured by the telomere. Okay, so this telomere is the one involving in aging, cancer, cell lifespan. Everything will be decided by the presence or absence of this telomere. Okay, see here, telomerase activity is linked with the cell division potential. Of course, it is right. Okay, so telomerase, what is telomerase? The enzyme which is present in telomere is called as a telomerase. Okay, the enzyme which is adding the repetitive DNA sequences in telomere is telomerase. Okay, now see here, so now you should know what is the shape of the chromosome based on the position of a centromere. See here, so what do we have studied? So this is what a chromosome, isn't it? This is a chromosome and this is the centromere. Based on the position of a centromere, we are having a different types of chromosome based on the position of a centromere. If the centromere is present at the center and we are having a V-shaped chromosome, then we can call them as a metacentric. Okay, if it is an L shape, okay, it will be, if it is an L shape, it is called as a submetacentric. If the P arm is very short and they are having a very long arm, then it is called as an acrocentric, which means towards the end. It is not exactly at the end. If the centromere is present exactly at the end, we are calling them as a telocentric. Near the end terminal, which is near the terminal end, we are calling them as the acrocentric. Okay, now see here, the centromere position will be terminal, then we are calling them as the telocentric, and the shape will be rod shape. If the centromere is present in near the terminal, we are calling them as an acrocentric, and again it is called as a rod shape. Okay, if it is present in the subterminal, which means it is an L shape, then it is called as a metacentric L shape. Now exactly if it is present at the center, that is called as a metacentric, which is in V shape. So this is what a type of chromosome, shape of a chromosome based on the position of a centromere, right? Now, functional classification of a chromosomes. Chromosomes can be classified based on the function. Okay, so totally we are having 46 chromosomes. In the 46 chromosome, if we set in a pairs, that is 23 pairs of chromosomes. Okay, so if it is in pairs, 23 pairs of chromosome, 22 pairs is considered as an autosome, and one pair we are calling them as a sex chromosome or allosome. If it is a 46 chromosome, if it is in numbers, 46 chromosome, in this 46 chromosome, 44 are considered as an autosome and 2 are considered as a sex chromosome or allosomes. Okay, autosome is considered somatic chromosome, that is body chromosomes. Now see here, autosomes control the somatic bodily characteristic features that is present in both the sexes, and in humans we are having 44 autosomes and which is existing 22 pairs of chromosomes. Now what is with respect to the sex chromosome? Sex chromosome determines the sex of an individual. XX is considered as a female and XY is considered as a male. Okay, now, so this is the area where we can expect a question short, okay. Special type of chromosomes. We are having some special type of chromosomes and we are calling them as a giant chromosomes. So we are having two different types, special type of chromosome. One is polythene chromosome and another one is lamb brush chromosomes. So first we are going to discuss about this polythene chromosome. So this is what a polythene chromosomes. This is found in the salivary glands of Drosophila larva. Okay, salivary glands. So this is their statement where we can expect in the questions. Okay, this is found in salivary glands of Drosophila. Okay, and this is first observed in the Balbiani rings. Once again, see here, this polythene chromosome, this is found in the salivary glands of Drosophila larva. Okay, salivary glands of Drosophila larva. This is first observed by the E.G. Balbiani. Okay, so that is why the rings which is present in this chromosome are called as the Balbiani rings, as it is discovered by the Balbiani scientist. Okay, so this is important thing, how it is getting formed. See, they are having a repeated DNA replication without nuclear division. The nucleus is not dividing, but instead the DNA is getting replicated repeatedly, but no nuclear division. Okay, so this leads to the formation of a giant chromosome. Okay, so why it is very giant? Because the nucleus is not dividing, but instead the DNA is getting replicated continuously, but the chromosomes, that's why the chromosomes looks bigger. Okay. Now, see what are all the features? It will be visible under the light microscope. They are having a dark band and light interbands. Okay, see here they are having a dark bands and dark bands. Between the dark bands we can see the light interbands. So 95% of DNA in bands only and 5% of DNA in the interbands. So dark bands is predominant in case of polythene chromosomes. Now what is the Balbiani rings present in the polythene chromosome? That is called as a chromosome puff. See here, very large puffs seen in the chironomous larvae and sites of intense RNA synthesis. This is the area, this is the Balbiani ring area where we can see the intense RNA synthesis is taking place in the Balbiani ring. So this is why we are also called as the chromosomal puffs. And polythene chromosomes are generally genetically active. This is an important statement, okay, because this is the site of RNA synthesis, so it is genetically active. So this we can expect in the exam. And the lamb brush chromosome, so this is what a lamb brush chromosomes. It occurs during the diploting stage of the prophase 1 in the meiosis. See, we are having a meiosis and mitosis. So in the meiosis we are having prophase, metaphase, anaphase and telophase. In the prophase we are having liptotin, zygotin, dipacketin and diplotin and diakinesis. So this lamb brush chromosome will be occurs during the diplotin stage of prophase 1 of meiosis. Okay, so where it is present, so in case of polythene chromosome it will be found in the salivary glands of the Drosophila larva, but in the lamb brush chromosomes this is found in the oocytes of the salamander, giant nucleus of the acetabularia. Okay, so this is very important. Okay, so it is first observed by the Fleming in 1882. Now, how its structure will look like? There is a central chromosomal axis. Okay, so there is a loop-like structure. So there is a lateral loops of numerous lateral loops of DNA will be seen in the chromosomes. Okay, what is its significance? See, these are all the lateral loops represent the sites of active RNA transcription. So this is the area, again it will be active, genetically active, where we can see the RNA transcription will be taking place. This indicates high gene activity during the oocyte growth. Okay, so during oocyte growth we can see this lamb brush chromosome. Why? So there we can see high gene activity. Okay, so this is what This is what with respect to this Lamb-Brush chromosome and the polyethylene chromosome. And important one-liners are the chromosome term was proposed by Waldeyer, and who said the chromosomes are the carrier of a genes, that is Bridges. And the nucleolar organizer region is the region where we can see the rRNA synthesis will be taking place, ribosomal RNA synthesis. SAT chromosome, that is satellite chromosomes, will be present from the, it is a small segment which is separated from the secondary construction as SAT chromosome. Now polyethylene chromosome will be seen in the salivary glands of Drosophila larva, and in the Lamb-Brush chromosome will be seen in the diplotin stage of prophase 1 of meiosis in the oocyte. Now, telomeres. In the telomere, we have studied about the telomere. Telomere is made up of a repeated division, and that repeated sequence is TTA, GGG. In case of humans and Neurospora crassa. Okay. So, these are all an important one. Let's how the exam may trap you. Okay. See, the chromosomes occurs in prokaryotes. No chromosome will occurs in the eukaryotes. And nucleolus forms at the centromere. No nucleus forms at nucleolar organizer region. And polyethylene chromosome occurs in meiosis. No, it is not in a meiosis. And Lamb-Brush chromosome occurs in a mitosis. No, it is a false statement. It is occurs in a meiosis. Now comes the MCQs. Okay. So the first question, the nucleus was first described by the Robert Brown in 1831. Which structure is directly connects the nuclear envelope with the endoplasmic reticulum. Okay, which structure directly connects the nuclear envelope. So which structure? That is obviously the outer nuclear membrane. So we have studied about that. So which of the following function is primarily associated with the nucleus? Here they are asking with respect to the functions associated with the nucleolus. So that is exactly the ribosome biogenesis, not the protein synthesis. Okay. Which type of chromatin is transcriptionally active? Transcriptionally active. See, I have forgot to mention that. See, euchromatin and heterochromatin. Okay. See, just make a note of it. Okay. See, euchromatin and heterochromatin. Heterochromatin are transcriptionally inactive. Euchromatin are transcriptionally active. Transcriptionally active means protein synthesis will be taking place in the euchromatin and it is not taking place in the heterochromatin. Why? See, say for an example, heterochromatin will be look like this. Okay, euchromatin will be look like this. Okay, see here, the regions present in the euchromatin is clearly visible when compared to the heterochromatin. Heterochromatin, it is not clearly visible. The sequence present in the heterochromatin, it is not clearly visible. But the sequence present in the euchromatin is clearly visible. So, it will be easily identified by the RNA polymerase. So, the transcription is taking place. That is why it is transcriptionally active. Okay, so but here it is not clearly visible, so RNA polymerase can't find the promoter region, so it can't bind, so the protein synthesis is not taking place. So that is what transcriptionally inactive. Okay, and based on the stain is here, it will be darkly stained. It is lightly stained. Okay, so this is the major area where we can expect a question. Now, which type of chromatin is transcriptionally active? That is obviously the euchromatin. Okay. So now, and also, so like when we say which is loosely packed and which is tightly packed, when seeing this, euchromatin is loosely packed and this heterochromatin is tightly packed. Okay. Now, nuclear pores allows the transport of macromolecules. Nuclear pores allows the transport of macromolecules primarily by how the transport of macromolecules will be taking place in case of nuclear pores. Okay, that is exactly the signal mediated active transport. Okay, so which of the following cells is anucleate in mammals? Okay, we have studied which is anucleate in mammals, that is none other than RBC. Heterochromatin is generally characterized by heterochromatin will be looks like this. So it is dense packing and is transcriptional inactivity. Okay, so that is the right answer. Which event occurs during mitosis with respect to the nucleus? During mitosis, which event occurs? Nucleolus enlarges. Nuclear envelope remains intact, nuclear envelope breaks down, chromatin remains uncondensed. Which is going to take place? Nuclear envelopes breaks down in case of mitosis. Now, which statement correctly differentiates a nucleus from nucleoid? What is the difference between nucleus and nucleoid? Nucleoid contains a linear DNA. Of course, it is right. Nucleus lacks histone proteins. It is wrong. Nucleoid is the one lacking the histone proteins. Nucleoid is membrane bound. No, nucleoid is non-membrane bound. Nucleus contains a histone associated DNA. Of course, it is a yes. See here the question is asking how, which statement is correctly differentiating a nucleus from a nucleoid. Okay, so nucleoid contains a linear DNA. It is wrong statement. It is a circular DNA. So the correct answer will be option D. Right now. The final question, which of the following is not a function of a nucleus? Regulating a cell cycle, of course it is a function of nucleus. Control of gene expression, synthesis of a ribosomal RNA, ATP protection. So which is what are not a function of a nucleus? Obviously it will be the ATP protection. So that's all about the nucleus. We have completely discussed about the nucleus with respect to the CUET exam. Okay. Thank you so much. We'll meet you in the next class with us another cell organelle. Okay.